you're absolutely right, Robin. You can increase growth if you increase temperature to a certain point. But here we're really talking about limiting growth or reducing growth. So when might that occur?
It kills them.
Say again?
It can kill them by denaturing the proteins in their system.
Yeah, Daniel. So if you exceed their optimal growth temperatures, right, then oftentimes what's happening is proteins are denaturing. If it gets really hot, DNA can unwind, right? Cells can lyse, these kinds of things. So it's a fine line because increasing temperature can actually speed metabolism, speed growth. But then if you exceed that, then it can definitely be in this category of limiting. So all three of these were correct answers, and so E was the best answer. Does everybody see that now? Awesome. Okay. So let's talk about some of these conditions. I've introduced a few of these in this, but there's certainly more, okay? And so I'd like us to explore it. So abiotic factors are factors that environmental conditions are things that are not consumed biologically, okay? So what are the factors out there that might be modified by growth or organisms, but pretty much they're abiotically driven? So for example, bacteria and archaea can survive in the deep subsurface, kilometers down below the surface of the earth. And we're even gonna read the Chivian paper in a few weeks where we talk about that, okay? So what I want you to do is we're gonna have another quick breakout session. It won't be quite as long as last time's. But I'd like you to just come up with a group list of the conditions you can think of that might shape your sort of the growth or impact these environmental conditions that make these impacts. Okay, so we're gonna have 6 groups again. And let me just make sure. Oops, hang on. What did I do? Oops. Sorry. All right, go ahead and accept your room and just come up with a list as a group of the conditions that you can think of. And then we'll come back and share. All right, welcome back. Hopefully you guys had enough time, but even if each group didn't come up with an exhaustive list, I hope collectively we can go ahead and come up with that. So somebody want to share maybe one of the traits or the conditions that you were talking about in your group?
One of the conditions we talked about is pH.
Okay, pH, I like that. Somebody else?
We talked about pressure, how it might change the environmental conditions for a bacteria.
Okay. Somebody else?
We talked
You said
about
light
temperature.
Go ahead, Sandra. You said
We
temperature?
talked about temperature as well as osmotic pressure.
Okay. And then who else?
Oh, that was
We talked about sunlight exposure.
Sunlight. Okay. Thank you, Rika.
We talked about salinity.
Okay. I think somebody else was trying to speak, maybe.
Oh, I was gonna say light exposure as well.
Okay,
Yeah.
that's cool. Thank you, Hector.
Oxygen requirement.
All right, who was that?
Lillian.
Lillian? Okay, thanks, Lillian. All right, great.
Resource availability, like nutrients.
Okay, so that is going to be, you're jumping the gun on our next thing, because remember, these are things that are abiotic factors that are really not going to be consumed, but we're going to come back to that. So put a pin in that, okay? And we will definitely talk about those. All right, well, I think you guys did a great job. So you pretty much came up with everything on this list. Oxygen is a little bit tricky because oxygen can technically be locally consumed, but certainly the result of that in terms of the oxidation state is something that we typically think of as a condition. You guys got the temperature, pH, pressure, salinity, and light, right? So there's UV components of that, which can be very damaging, as we've talked about, as well as other, of course, visible light can be really important for photosynthetic organisms or organisms that have light absorbing pigments as well. And typically we're not going to exhaust the sun, right? So, yeah.
Professor,
Yes.
can altitude be a good condition?
So we will actually touch on this a little bit when we talk about habitats. Altitude per se, I don't think would be a condition, but altitude is certainly associated with typically low temperatures, right? So in that way, it might indirectly affect things. And we're going to talk about dispersal and aerosolization and movement of things in the atmosphere when we talk about our habitats. So that's an interesting point to make, yeah.
Got it, thank you.
Yep. All right. Okay. How is this going forward? Okay, so this has been studied for a very long time. And so there's often these theorems or postulates or laws. We don't have a lot of them in ecology in contrast with like physics and things like that. But there definitely are some things. And so one of them related to conditions is Shelford's law of tolerance. And what Shelford said was that the environmental parameters or bounds, right? So the range or the extent of what these various conditions are will determine both the abundance and the distribution of organisms. And that should sound kind of familiar because when did we talk about abundance and distribution on the first day? What context did we use that in? Do you remember?
We're defining ecology.
Right. So essentially environmental parameters they determine the ecology of organisms. One of the things we need to think about though is in an organism's lifetime, even a microorganism's lifetime, we're really looking at averages over time. Think about it. You know, it may be in the 50s today, And, 3 months from now, it may be in the 90s, or even 2 weeks from now, because, this is California. But really, and so that is, right, weather versus climate. And this is something that a lot of people, I think, are struggling with in terms of climate change and these kinds of concepts. For an organism, when we talk about tolerances, we're really talking about averaging it over your life. You may die if you're exposed for a brief time to very extreme temperatures, say. But in general, what you're looking for is how these parameters on average impact you. And so organisms will have ranges that they can live in, that they have evolved and adapted as populations over time to live in. But what goes on a single day may not be the main driver of the sort of trajectory of those organisms in terms of their growth and survival. The other thing to think about is interactivity across conditions. So we're never exposed to just one, right? We're going to be exposed to a combination of temperatures and pressures and salinities and oxygen and right? And these often interplay with each other. And so organisms have to have evolved across those interplays. And this is kind of related to what I was saying earlier, where no single organism or species has the same niche. And that's because the way all of these things interact and their tolerances or preferences for these varies from species to species or population to population. And relating back to kind of what we talked about with our growth curves, we're going to see huge differences in how the effects of these environmental parameters have in the lab versus in the field. If I take a pure culture and I measure the growth potential of that pure culture, let's say over a range of temperatures, and these are actually experiments we do in my own lab, right? I can come up with a growth curve and say, oh, the optimal temperature for growth is 42 degrees for that species or 28 degrees for that species. But remember, if you're out in the real world, the optimal growth temperature may not be at the same temperature because of all the interactions with all these other conditions that are going on. Something to think about. The other thing is we tend to take a very anthropocentric or human-focused view. And so we like to use the term extremophiles, right? When we talk about microorganisms that live on high temperature, low temperature, or high pressure. But remember, those microbes that live under those conditions, they're adapted to those. And so they would actually find our optimal conditions of, 28 degrees Celsius to be very stressful. If you're optimally designed to live in 80 degrees Celsius, you would maybe die if you were put at room temperature, right? So we need to keep that in mind when we talk about stress, okay? What is stressful for us is likely very different than what is stressful for microbes out there in the environment. All right, so we have some terms that relate to sort of the tolerances versus really liking or being optimally adapted to it. So we have these two terms, acclimation and adaptation. Does anybody know the difference between those two definitions? What does it mean to acclimate to something?
Getting used to the environment.
Okay. And so what's the time frame of acclimation usually?
2
What
weeks.
would you say? Ballpark.
2 weeks for a human.
Okay. So like for humans, like let's say you are going to altitude. Here's altitude again. You go to altitude and it takes your body a while to acclimate to those conditions, right? Microbes are going to be no different. But the key thing there is it's over relatively short periods of time. Adaptation, how does that differ from acclimation?
Adaptation is permanent.
Okay. And how so? What are we really talking about when we talk about adaptation?
Well, adaptation is basically like physical and chemical changes that organism basically goes through in order to adjust to that environment on a more permanent basis.
Well, and actually, I think it's even a little more permanent than what you're alluding to, Robin, in the sense of when we use the term adaptation, we're really talking about the process of evolution, right? And so it's not necessarily you and me adapting, but it's our offspring or our offspring's offsprings or whatever. So adaptation is really something that occurs over longer periods of time and requires essentially genetic changes or changes in the allele frequencies of a population, for example. So acclimation is something that can happen in an organism's lifetime, whereas adaptation is something that really can only occur due to differential survival and reproduction, which is essentially how evolution works. So for example, photoacclimation, a photosynthetic organism can adapt or can acclimate, excuse me, to low light conditions by producing more chlorophyll. So let's say a cyanobacterium is floating in the ocean and it gets lower in the water column than it was the day prior. Well, less light is going to get down to where it is, and so it may produce more chlorophyll to adapt or to acclimate. See, I mix these up too, so it's acclimate to that lower light condition. But if you brought it back up to the surface, then it would just reduce the chlorophyll, right? And it's not adaptation in the sense that it's the same organism that can make changes in proteins or pigments or things like that. Adaptation would be if there's a low light variant that only produces a certain amount of chlorophyll. And that's because the ones that produced more chlorophyll died or didn't reproduce as quickly. So do you see the distinction there between acclimation and adaptation?
Professor?
Yes.
So can you say that acclimation is a temporary physiological change in an organism?
Yeah, I think that's a fair definition because it doesn't have to last forever.
And there are no genetic changes in
Correct. Correct. Other terms. So, you know, you've heard the term thermophile before, right? And when we talk about a thermophile, you're really talking about an organism that is adapted over many generations, over long periods of time, millions to billions of years, right? To living at a higher temperature or a lower temperature or a higher salinity or whatever. So if you're a thermophile, you're adapted to living at high temperatures, okay? That contrasts with, so that's adaptation. Tolerance, however, is, it can be adaptive, but it also can be something where acclimation can occur. So an organism can tolerate a certain set of conditions, but it's not their ideal growth condition, okay? So for example, psychrophiles, which are the opposite of thermophiles, right? They grow under low temperatures. Their optimal growth temperature will be less than 15 degrees centigrade, okay? A psychrotolerant organism actually will grow higher than that. So it'll grow really well at 20, which is about room temperature, right? However, if you take it down to the range of 10 or 15 degrees, it still grows. So it's not dead, it's not super unhappy, it's just less happy. It's less optimal for it at 10 degrees than it was at 20 degrees. So this we would call a psychrotolerant or low temperature tolerant organism. Okay, does that make sense? So we use these terms pretty broadly. Thermophiles typically can't grow below 50 degrees. This would be a classification definition we have given to thermophiles. Remember, 37 is body temperature, right? Something like E. coli living at human body temperature is not a thermophile. they'd be referred to as a mesophile. They like it in the middle, meso, not too cold, not too hot. It turns out that thermophily, actually, because there is really a very large, hang on a second, let me let somebody in. Okay. Thermophily, we have some that grow above 50, and then we have some that grow even higher. Okay, and we'll come back to that. Another set of terms is steno versus uri. So if you are stenohaline, you like a very narrow range of salt. Okay. If you are urihaline, then you tolerate or do well under a broad range of salinities. Okay. Or you could be stenothermal, right? I grow very well. You know, this organism grows at between 55 and 60 degrees, and that's it. So if you are a tolerant organism like this, you're more likely to be uri for whatever that is, because you can tolerate a broader range, even if it's not your optimum. Does that make sense? Okay. All right, so that's audicology. Conditions impacting organisms, they don't really have much to do to change it. They just have to respond to it, and they adapt to it over generation after generation, changing the population to be adapted to a given set of conditions. The field of synecology takes into account how these populations interact, right? And that concept of competition that we mentioned earlier, right? And so those interactions are often going to be mediated by resources, which can be consumed. And so these are not abiotic factors, these are biologically mediated factors, okay? So synecology really focuses on the outcomes of those kinds of interactions. And both audicology and synecology play a very important role in determining when and where organisms grow and survive, et cetera. All right, so now let's think about resources. These are different than the conditions we talked about. So I'm gonna open up the rooms again, and you'll have a couple-minute breakout, and let's come up with resources that you can think about. So Kaden, can you accept the breakout room? Sorry, Kaden, I didn't realize because you had been dropped from the Zoom and then I brought you back in, you weren't automatically assigned to the breakouts again. Hopefully you were able to join the end there. All right, so maybe somebody who hasn't said anything yet today want to volunteer to share a resource?
So chemical sources being like food or so it's kind of like a double-handed one. So food resources, but also we would say location because they want to be in like the region with like the most resources for the organism to consume. So there'd be a competition of space and food.
Okay, so space can be competed for and food. Okay, those can both play a role. And you can imagine, can somebody give us an example of where space competition, you might have even seen that, like in the lab even?
Amy gave a good example. Well, not necessarily in the lab, but of like microorganisms on our skin.
Okay.
So like this competition from invading pathogens. So you have like
Yeah.
Staph aureus on our skin.
Okay.
Staph epidermis.
Cool. Yeah, no, that sounds like a good example. Even just a colony on a plate, right? 2 colonies can be competing for that. So, all right. Somebody else want to share?
Our group said nutrients like carbon and nitrogen.
Yep. And so that's kind of getting to what Victor said in terms of food, but it's giving more specifics. So definitely carbon is going to be our, sort of main building block, but as we already alluded to, nitrogen plays an important role. Absolutely. Anybody else?
Oxygen.
Oxygen. Okay. And was that nitrogen or?
Light.
Okay. So light can be sort of, right? So photosynthetic organisms kind of absorb light. And so if you're an organism up here absorbing light, then those photons of light may not get down below. So light can be a source of competition, even though we also listed like UV radiation as a condition. So that one is kind of one of those squishy ones. But yes, oxygen definitely. So we said that redox is more of a condition, but of course, redox and oxygen are intimately tied together, oxidants and reductants, right? But oxygen can definitely be locally consumed. And so if you dig into the mud in the bottom of the ocean, right, it's usually anoxic because any oxygen that makes its way into those sediments will be rapidly consumed by microbes living there that will respire it and remove it from the system. Okay. Anybody else have anything? Okay. I think we covered most of what I have. I don't think we hit this one though. Where might you compete for water? What kinds of organisms might end up competing for water? Or in what habitat?
Dry habitats?
Yeah, so like the desert, right?
Mm-hmm.
So a desert microorganism may not see water very often. And so at this time of year, when it rains in the desert, this might be their time to be able to use that resource. Hopefully they have these other things like nutrients available. They can reproduce really quickly, and then they may have to go back into dormancy for another 10 months or 10 years or 100 years, right? Depends on where they are. There's some very dry places in places like Antarctica, in the Andes Mountains, and in some places where there's rain shadows, where rain is really, really rare. So water can also be something. All right, cool. So just like we had Shelford's law of tolerance, we have Liebig's law of the minimum. So this is another one of those. eminent scholars that came up with his own law. And so what Liebig said was that we're limiting nutrients. That's what's going to drive and determine microbial growth or biomass. And this gets back to our growth curve that we talked about before. The reason it went into stationary, we said, was because it ran out of something. And you don't have to run out of everything. You only have to run out of one thing, and that's referred to as the limiting nutrient. So when an organism or a population of organisms like microbes is growing, they're going along, and then something runs out. And this is typically going to be a major driver in the cessation or the halting of microbial growth and the total level of biomass they achieve, how high up on that curve they get. So it turns out that this is really important in some places, like with algae found in the ocean. And it's been quite well studied by somebody named Redfield. So Redfield basically ground up algae and analyzed them and realized that they have about 100 parts carbon to 16 parts nitrogen to 1 part phosphorus. And Redfield came up with this ratio and said that because this is what is necessary for there to be cells of these algae, that this is the ratio that would be needed for them to grow. And the good thing for algae is that carbon is accomplished or achieved or obtained via carbon fixation, right? So they have pretty good access to this. And so what Redfield said was either nitrogen or phosphorus is going to be limiting. And this is an interesting concept because you may look at this and say, well, there's 16 nitrogen and there's 1 phosphorus needed. So nitrogen is going to always be the thing that's limiting. And that's actually fairly commonly true in the ocean, but it turns out that in freshwater habitats, and we're going to talk about some of these next week, phosphorus turns out to be, and the key thing is that it's the ratio. So even if you have a bunch of nitrogen, if the ratio of carbon to nitrogen or nitrogen to phosphorus differs, then it can stop growth. So even though you only need 1 phosphate for every 16 nitrogen and 100 carbon, you can still end up being phosphate limited. And that's often the case in freshwater lakes and those kinds of habitats. But looking at this ratio has sort of been kind of a classical thing in looking at limiting nutrients. I will say, however, that we've come up with a more sophisticated view. And as I mentioned earlier, you know how we're looking at the averages of conditions impacting organisms? We now think that the Redfield ratio is really an average across many organisms at any given time, but if you, or over time, but if you look at a specific snapshot of carbon, nitrogen, and phosphorus at any one time, you can actually be out of so-called Redfield ratio and still have growth. So it's a little more complicated than they really came up with, but it's a good way of us to think about these nutrients, okay? And one thing we're going to talk about a lot this semester is the impact of fertilizers and human impacts on environments. As I hope you're aware, fertilizer, right? It turns out we need nitrogen and phosphorus in large amounts to grow plants. But the same nitrogen and phosphorus that are required by photosynthetic plants have a huge impact on photosynthetic algae. And when farmers put nitrate on their farms or phosphorus on their, on their farms for their crops, when it rains, that gets flushed into the stream near their farm, runs into a river, and ultimately finds its way to the oceans. And many of the world's oceans are unfortunately majorly impacted by fertilizer pollution. And a lot of water bodies, freshwater and oceanic, are what we call eutrophic, or having too much nutrients, too many nutrients, especially nitrogen and phosphorus. What this creates is an environment where algae will bloom in huge quantities, which you might say, oh, well, that's good. They're photosynthetic. They're producing carbon and releasing oxygen. That's good for us. But the problem is when the algal bloom runs out of something, it dies. it falls to the bottom of the ocean or the lake or river. And then those microbes living down there will decompose the dead algae. And as you know, respiration consumes oxygen. And so what happens is the demand for oxygen goes way up. The so-called biochemical oxygen demand is very high when these blooms fall to the bottom of the ocean or fall to the bottom of a lake. And they create oxygen-free dead zones. And so if you look at the mouth of the Mississippi River, the largest river in the United States, right, flows out lots of agriculture all the way up into Canada and Minnesota. That's where the Mississippi starts. And then it goes all the way down to New Orleans in Louisiana. And then it flows out into the Gulf of Mexico. And there's, because of all of that nutrient pollution, there's a huge dead zone that forms, particularly in the spring and summer when the algae have plenty of sunlight and it gets warm temperatures. And what happens is the oxygen gets consumed and it kills the fish. So it's really bad for the fisheries in that area. And this problem is becoming more and more prevalent all over the world, even on our own coast. So for example, the Columbia River that flows in near Portland in the northwest also has lesser dead zones. Something to think about. And it really, when we throw these nutrient levels out of whack of what would normally happen, right, out of the normal ratios, that's when we would end up with trouble. Interestingly, in the ocean, far from the coasts, out in the middle of the ocean, it turns out that nitrogen and phosphorus often aren't the limiting nutrient. It's often iron. So iron is only required as a trace element in very small amounts. But it turns out that this way out there, nutrients are so low that iron can be limiting. So one way that we need to think about this is, how do the microbes deal with low nutrient availability? And we have some more definitions to go by. Organisms are adapted to where they live. So if you live near the coasts and near where the rivers are bringing in lots of nutrients and there's lots of carbon available, you'd be referred to as a copiotroph. Maybe you've heard the term copious, where you have lots or an abundance of things. Oligotrophs are organisms adapted to living under low nutrient conditions. So that's some terminology that we use. We also have some other terms. So we tend to think of culturable organisms, right? Microbes that are culturable. And we talked about already that the majority of microbes have never been cultured before. And one of the terms that comes up for that explains it is that there are oftentimes in environments, there are organisms that are viable, but we don't know how to culture them. And so they've been given this VBNC designation, and you'll see this in some literature, where they're referred to as they're alive, but we don't know how to culture them. And so we can't really study them as well. Now it's interesting because if a microbe is living in an environment where nutrients become limiting, and that's very commonly the case, they may have to go dormant, as we talked about. And so Dr. Epstein from Harvard in 2009 came up with this study where he postulated a scout hypothesis. So what this predicts is that microbial populations will go dormant, but then every once in a while, one cell will wake up. And it's a scout. It's basically sensing the environment and asking the question, are there nutrients now? Or are the conditions right now? And if they are, it'll grow and form a new population. If they aren't, they'll die and be sacrificed. But the rest of the colony or the population is still dormant. So this will episodically happen and can explain how these organisms respond to the fluctuations over long periods of time. This may take days, weeks, months, years, decades, centuries even, given the different circumstances. The other things microbes can do is they can scavenge nutrients. So they can produce molecules. One of them I mentioned, iron, can be limiting in the Central Ocean. Siderophores are actually extracellular compounds that can be released by cells, which are chelators. You guys remember from chemistry, what does a chelator do? Who remembers?
Removes heavy metals.
Well, it can remove them, but basically, how does it remove them? Do you remember, Robin?
I don't.
No. Does somebody remember? So a chelator binds, right? And so that's how it's removing. If it binds it and precipitates, it might fall out of solution. I think that's maybe what you were getting at. In this case, actually, though, they bind and they don't make them precipitate, they make them more soluble. So these are binding and solubilizing, making them easier to take up into the cells. Another example are alkaline phosphatases. It turns out that out in the environment, most phosphorus is not in the form of Pt.2 phosphate that you've used in chemistry class. So oftentimes it'll be in organic forms from other dead life material out there, other biomass. And so alkaline phosphatases cleave the phosphate off of organic, phosphorus-containing organic molecules, liberating the phosphate, which is then easily taken up by most cells. And then we already had this mentioned in the chat, nitrogenases are specifically evolved enzymes in certain groups, mainly of bacteria, that can fix atmospheric nitrogen and turn it into usable ammonium inside the cell. And we're going to talk a lot more about nitrogenases later. All right, we have run over time, so I will finish this out next time. Are there any questions?
Could you just go back to that last slide for me, please? Perfect, thank you.